Okay, Assalamualaikum warahmatullahi wabarakatuh. So we continue with lecture number 5 for chapter number 2. So structure of protein and nucleic acid. Okay. So first we see the protein. Okay, protein structure. Protein are extraordinarily complex molecule. So we can see here in this picture, it's very complex molecules containing of some carbon, hydrogen, nitrogen, sulfur, and so on. Okay. So these are one uh, image, and this another image or type of image. Okay, so we can see that these are the protein. Okay, so the protein structure, level of structure of organization of protein. So we have four structures. So the first one is primary structure, the second one is secondary, tertiary, and quaternary structure. Okay, so these are the primary structure, the sequence of amino acid residue, lysine, valine, adenosine, valine, aspartame. So I'm making the bond. Secondary structure, you can see that here is the linear line, but in the real one, we have zigzag, 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 depend on the direction. And then the three structure, okay, so here you can see that this adding to this part. Okay, so we get a three-dimensional structure of the entire polypeptide. Okay, the side chain. Then the quaternary structure, so you can see here, these are one polypeptide, these are one protein, this one protein, this one protein, combined, making the this polypeptide, making the protein. Here's first we see the primary structure. So every polypeptide has a specific amino acid sequence. Okay, specific sequence. The interaction between amino acid residue determine the protein three-dimensional structure and its functional role and relation to the other protein. Okay, so this interaction, you can see that determine the protein three-dimensional structure and its functional role. Okay, so when this the amino acid uh, amino acid chain with another amino acid, so it will have a different functional role. So here we see the tyrosine, lysine, phenylalanine, leucine. Okay, so these are one polypeptide. Okay, so these are residue. So this amino terminal residue, and here we have carboxyl terminal residue. Okay, so we have this. Okay, next we're going to see the secondary structure. So secondary structure is polypeptide chain can fall into regular structure. Okay, and the most commonly observed type of secondary structure are the alpha helix and beta pleated sheet. Okay, so you see alpha helix. The alpha helix is stabilized by hydrogen bond between the N. These are just example NH and CO group of the main chain. So you can see here, this is one. This so this one. So we have the hydrogen bond between this uh, nitrogen and the these are oxygen, and these are the hydrogen of this nitrogen. Okay, so we can, if we say that these are H, the green is hydrogen, or no, this green is the nitrogen. Okay, so we can see that these are the value. Okay, so if we make this type of swirl, okay, putaran. Okay, so we can have this alpha helix. Okay, so these are very long polypeptide, but because of this hydrogen bond, so it can become this figure or this configuration. Okay, so here if we see again, so these are the linear amino acid, but when this hydrogen making the hydrogen bond with this oxygen, so it will fix the the polypeptide. Okay, making this become the alpha helix. Okay, again this oxygen and this hydrogen, so making the hydrogen bond, making this twist. Okay, so next is the beta pleated. So beta sheet is composed of two or more polypeptide chain called beta strand, and beta strand is almost fully extended rather than being tightly coiled as in the alpha helix. Okay, this are alpha. Okay, alpha helix. And the distance between adjacent amino acid along the beta strand is approximately 3.5 angstrom. Okay, so you can see here, these are nearly straight line, straight, but we have two strand. Okay, so these are one polypeptide and this one polypeptide, and here is making the hydrogen bond here, hydrogen bond here, and hydrogen bond here, and hydrogen bond here. Okay, so you can see in this case, okay, the atom is displaced at the some point. Okay, these are anti-parallel. One goes here, there, and one goes. Okay, so you can see these are, let's say these are the nitrogen, and here we have this nitrogen. Okay, okay, and for parallel, okay, so we have the same structure, blue and blue, black and black. Here this black and blue, here black and black. So making the hydrogen bond here, here, and here. Okay, so from this alpha helix and beta sheet, okay, so we can produce a super secondary structure. So here many globular protein contain combination of alpha helix and beta pleated sheet secondary structure. So here you can see that this one beta alpha beta. So these are beta, these are beta, this is alpha, and these are beta middle. Okay, alpha alpha, beta viral here, and here Greek T. So we have this super secondary structure. So next we go to the tertiary structure. So we're going to have unique three-dimensional conformation that globular protein assume as they fall into their native structure and proteotic group. Okay, so when it become this structure, it will be biologically active. Okay, so the tertiary structure has several important features. Many polypeptide fall in such a fashion that amino acid residues that are distant from each other in the primary structure come into close proximity. So sebagai contohnya C C15 let's see C15 dengan C150. Kalau ikut-ikut kedudukannya jauh, 15 dengan 150, but because of this folding, C15 will be near to the C150. Okay, and globular protein are compact because of efficient packing as the polypeptide fold. Okay, so boleh bagi macam COVID-19 punya contohnya lah, COVID-19 punya virus, macam bola ya. Large globular protein, those with more than 200 amino acid residue often contain super compact unit called structural domain. Okay, so some repeated domain. A number of related protein referred as modular or mosaic protein contain numerous duplicate or imperfect copies of one or more domain that are linked in series. Okay, so these are the three structure. So here, these are the interaction. Okay, so the interaction that maintain three structure. Okay, so here we have this hydrophobic interaction. Okay, so these are covalent bond, become a covalent bond. This from this R1 residue and to this R2 residue. Oh no, can see here CH3, so become a hydrophobic interaction. And this one is short bridge. Okay, so become 00HH. Okay, so this can be a, and this water bridge, oxygen, this water and this water can share the electron or the hydrogen, and these are just a hydrogen bond. Okay, so you can see that when we have the residue and the residue interact with other residue, so we can fold the long polypeptide into let's say this tertiary structure. So the next one is quaternary structure. Multiple polypeptide chain can assemble into a single protein. Okay, so this one, the green one is one, the yellow one polypeptide, the red one polypeptide, this yellow one polypeptide, this red one polypeptide. Okay, so you can combine together to make this quaternary structure. Okay, and this one is supposed to be in the direction, but here we have the energy of the bonding or the bending. Okay, the bending of the residue or the molecule. Okay, and the rotation of the bond. Okay, so we have the Ramanujan plot. The freedom of rotation about two bonds of each amino acid allow protein to fold in many different way. Okay, so kalau bond tu terlalu keras, so we cannot move around freely. Okay, macam kita punya apa siku ke bahu. Okay, kalau okay kita boleh buat macam-macam pergerakan. But kalau kita simen kita punya siku, kita simen kita punya bahu, so kita only boleh buat a certain pergerakan. Okay, so the angle rotation about the bond between the nitrogen atom and this is alpha carbon atom is called P, and the angle of rotation about the bond of the alpha carbon and the carbon atom called is psi. Okay, so here you can see that these are P and these are psi. Okay, so the rotation of the bond. So here we have the Ramanujan plot of the P against psi, value of possible conformation. So we have a lot of conformation, but these are the most probable configuration or conformation of this molecule. Okay, so here you can see that this one is higher than this. So kalau ada 100 molekul ni, okay, kalau ada 100 molekul yang ini yang sama, so most of them will be, let's say 70% akan berada di sini, and let's say 30% in this conformation. Okay, so from that energy plot, so we can see the distribution of the configuration. So the last one is structure of nucleic acid. Like in lecture number 2, nucleic acid exists in cell in two varieties: DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Okay, so it's function as the carrier of genetic information by virtue of their structure. And the sequence of base usually characterize a nucleic acid and is a form of linear information. Okay, so this were sugar, phosphate, base, base, and base. And DNA is more stable and better suited for information storage. Okay, so our DNA tak akan berubah ataupun berubah sedikit je bila diturunkan ke anak. RNA less stable and more useful in information transfer as a messenger, a translator and a static machine. So RNA ni kata nak buat signal to the brain, for example. Okay, so DNA and RNA differ in the sugar component and one of the base. Okay, so here we can see that these are DNA, the base, mungkin ada beza dengan base, and sugar component. So here you can see that for DNA, these are hydrogen, but for RNA, these are the OH. Okay, and again, the backbone of the nucleic acid are formed by 3 to 5 photodiester linkage. Different in the sugar component of RNA is lighted in red. Okay, so hydrogen and OH. So these are the 3, and these are phosphate. These are sugar. These are phosphate. These are sugar. These are phosphate. Okay, and here we can see that if these are the DNA, okay, so we can cut this out. Let's say we can cut this out, and this A, T, okay, can be in this crucified form. Okay, so let's say this these are TGA. So this C will combine or connection with G. So here you can see C and G in this type of information. Okay, so we can get this type of form of our DNA. Okay, if we cleavage this or we cut this bond between this amino acid. Okay, so these are the amino acid. These are the same as like our COVID-19, the new variant, new mutation. Okay, so we have a different changing of this amino acid. Okay, so double helical structure. So what's an encrypt? So DNA double helix. Okay, so we have strand number 1 and strand number 2. So we can see that it will do the hydrogen bond, the phosphate bond, sugar, these are phosphate, phosphor bond, and some other bond. So we can get this DNA double helix. Okay, so every animal has a different DNA double helix, depend on the location of amino acid. But as we know that our DNA are nearly the same as the pig. RNA, the last one, I believe this one. Okay, so RNA, a single-stranded RNA molecule can fold back on itself to form a complex structure. Okay, so here you can see that these are the RNA molecule, so you can form this one strand, making so many type of configuration. Okay, so we can get this. So if we have the same amino acid, but with a different configuration or this folding, So we are going to have different information. Okay, let's say in this, form, it is, let's say it is on, and another form, it meaning that it is off. Okay, so let's say this RNA control the sensor, our heat sensor. Okay, so if in this form we feel the heat, but in another form we are not going to find, or we are not going to feel the heat. Okay, so these are the end for chapter number 2. Thank you.